We're going to start up with a Genetics 101 session, which I'm really excited to hear about. And for those that have their genetic reports or reports of their family members, it'd be great to, for you to get some more insight on what the report means, and because it is really challenging, even for, as Chris Will and others during yesterday's session was saying, it's even hard for medical professionals to interpret some of that. So hopefully Brad can shed some light on that, what it means, and what, and how best to interpret that, and what that means in terms of the molecular biology of it too, in terms of the protein, the RNA. So I'm just going to introduce Brad, because Brad is a great friend and also colleague of mine that I met quite a while ago when I was a PhD student. So embarrassingly, we're showing our age here. As a PhD student in Seattle, when he's been working with the Jane Foundation, it feels like forever now for me. So meeting up Brad, and over the years, I've got to know Brad as a person that's very generous with his time, whether it's explaining the genetics to other patients, either at conferences, either at on social media such as Facebook posts and other places, and also just doing it in such a way that's understandable, not in a way that scientists talk to each other, but in a way that patients would understand, and with such clarity, and also understand the importance of a genetic diagnosis and what it means in terms of patient care and what it means in the future in terms of treatment. So with that, I think he's done a fantastic job, and I think he's the perfect person to lead this session and present on it and then answer questions at the end. So take it away, Brad.
Okay. Thank you, Monkel. As Monkel said, I'm the work for the Jane Foundation in Seattle, and my official title is Director of Research and Advanced Diagnostics. Okay, so what this talk is going to cover is about what genes are, and well, basically the function of gene is to enable a cell to make a particular protein. So we'll go over how genes allow for proteins to be made, and then the structure of genes, and also types of mutations in genes. So mutations you can think of as spelling mistakes, and just as a spelling mistake in an important document could be a problem, a spelling mistake in DNA can be a problem in our case for some protein that our muscle needs to function. And I'm going to make it a little more comprehensible by using sentence analogies. I'll go over what that is, and then sort of how to understand mutations, and if you have a copy of your genetic report either here with you or at home, how to actually read that and know what it means in terms of what's actually happening with your DNA. Okay, so DNA is a series, is a double helix, and there are 4 different DNA bases which are abbreviated A, C, G, and T. And on one strand, an A on one strand is always paired with a T on the other. A G is always paired with a C on the other. So a gene is a particular region of DNA that contains the instructions for the series of amino acids which are to be to compose a given protein. Now we have about 26,000, or Monkel may actually have a more accurate number. We have a large number of genes in our bodies, but most of our genome is actually not genes. Most of it is DNA, which the function isn't entirely clear, or it has some other function. So one thing for genetic testing is, so whole genome sequencing is just to translate every base pair in all of your DNA. Exome sequencing is to just sequence all of the parts of your DNA that are actually part of a gene. Okay, so a gene is a recipe for making a specific protein. And proteins are chains of amino acids, and after the chain is put together, it can fold into various shapes, and essentially the shape determines the biological function. So you get amino acids from your diet. If you eat something from a chicken egg or a soybean or something that contains proteins, that has amino acids, your body breaks down the proteins from the soybean plant or the chicken into their component amino acids, and then your body reassembles those amino acids according to the instructions in the DNA to make its own proteins. So, yes, muscular dystrophies are often caused by protein deficiencies, but that doesn't mean that you just need to eat more protein. It's kind of like wanting to bake a cake if you've never baked a cake before, and you have all the ingredients, but you don't have a recipe and you don't know how. You aren't going to end up with a cake. Okay, so there are about 21 different amino acids that are incorporated into our proteins, and they all have a one-letter abbreviation or a three-letter abbreviation. And I'm expecting you to memorize this for the end of the talk. The other thing I will point out is that there's an X there, which is a stop. And what that means is that tells the cell, okay, this is the end of the protein sequence, we're done. But sometimes mutations can cause that to happen before the real end, and that's one type of mutation we'll go over. Okay, so how does the cell know from DNA what amino acid to put at the next place in the protein? Okay, so out of the 4 letters A and C, there's 64 different combinations of letters. And every one of those combinations either codes for a given amino acid or a stop. And you'll be expected to memorize all of this also. Okay, so as an example of how this actually goes into practice, so this is the dysferlin gene, which is absent or non-functional in LGMD2B. So it starts with this codon ATG, which codes for the amino acid that's abbreviated with an M. Okay, it turns out that every gene starts with an M. That's just the way biology works. Okay, so you have AGT that codes for an M, CTG that codes for an L, AGG that codes for an R, etc. And then you go on, there's a little over 2,000 amino acids in the protein, which means a little bit over 6,000 DNA codons in the coding part of the gene. And then finally you get to a TGA, which is a stop codon, which tells the cell, okay, we finally come to the end. We're done. Okay, we can now make the protein and fold it into its correct shape and have dysferlin, unless you have LGMD2B. Okay, so a genetic report, basically what it does is compares your DNA with a quote-unquote standard sequence. And there's going to be various differences, and they generally can fall into 3 categories. Okay, one is called a polymorphism, which means that it doesn't cause a problem. It's why, that's why some of us are taller than each other or have, different hair color or, whatever, trait that just makes us all unique individuals. Those are polymorphisms. They're not associated with disease. A mutation, on the other hand, is something that makes the gene not function correctly. And depending on whether the given gene and the disease associated with has dominant or recessive inheritance, sometimes you can have the disease by just having a mutation in one copy of your gene, or in a recessive disease you need to have it in both copies of your gene. These are on all the chromosomes that aren't sex chromosomes, which works out a little bit differently. Okay, and sometimes we see a DNA change that the genetic counselors and scientists, but Munkle is working on this, and you can ask him shortly. We aren't sure whether it's a polymorphism or a mutation, and that's called a variant of uncertain significance. That's basically genetic counselor speak for, well, this may cause a problem, we're not really sure though. Okay, so to illustrate different types of mutations, I could write long strings of A's, G's, C's, and T's, and you might need to go over to the coffee place to be able to stay awake. So I'm not going to do that. So I'm going to use a sentence analogy, and it's going to be a sentence that is all of 3-letter words. Why 3 letters? Because each DNA codon that specifies a given amino acid is a 3-letter word. Okay, so our example sentence is, the cat saw the rat, but not the bat. And then we have this word out at the end of the sentence, and rather than end our sentence with a period, we're going to use the code word out, which means, okay, this is the end of the sentence. That's going to be our stop codon in this analogy. The thing to know about DNA is we know where there's a little bit of a space between the and cat, so we know where to make it easier for us to read where one word, the breaks between words. DNA isn't like that. The cell just interprets every 3 DNA bases as a new word, and there's no spaces. Okay, so types of mutations with our okay, so the first thing is a polymorphism. And remember, this is one that doesn't cause disease. So an example of this is, okay, if we just spell cat with a K, okay, that's a little different. Your English teacher back in school might have, put a red mark on your thing, but everyone knows what that means. That's just different. Okay, and we're gonna sort of illustrate this with a protein shape as a little cloud, and so the protein basically still has the same shape as it did before. Okay, now this is the first type of mutation that is a real mutation and causes a problem. And this is called a nonsense mutation. Don't ask me why this is called nonsense. It never made a lot of intuitive sense to me, but this is what a nonsense mutation is. Okay, if we change the B in but to an O, then what happens? Well, now it spells out, and remember that was our code word for ending the sentence. Okay, so when our cell is, it's going to say, okay, the cat saw the rat, and then it's going to say, oh, okay, end of the sentence. Okay, so it ignores the, the part about, but not the bat. So we get only part of the protein. Now, depending on the protein, having half of the protein, may still work a little bit or pretty well or not at all. So this is a nonsense mutation. And so one example in real DNA, in a real gene, so this is in the dysferlin gene again, LGMD2B, at the DNA level, okay, the C basically explain in a genetic, this is how it would be written in a genetic report. The C indicates what's happening with the DNA bases. And so the P explains what's happening when it's translated into protein. Now you could, work it all, with the genetic code out yourself, but they normally, write both DNA and protein to make it a little easier to follow. Okay, so at the DNA level, you change base pair, the base 6,068 from a C to a T. And what does that do? That changes amino acid number 1905 from an R, which is, the amino acid is called arginine, to a stop. Okay, so that means that we're going to, okay, there's about 2,080 bases in DNA, so you're going to be missing about the last 175 of them. Okay, now the next type of mutation is called a missense mutation. And this generally means that you just substitute one DNA letter for another, but unlike spelling cat with a K, this does have an effect on the way that the protein looks. So if you say, if you change the R in rat to an H, then, okay, well, a rat and a hat are clearly different things. That changes the meaning of the word, or of the sentence. Okay, so your little protein cloud is going to be somewhat differently shaped. Now, a lot of times, substituting one amino acid for another is where a lot of these variants of uncertain significance are. It can be hard to figure out when they're just a polymorphism, harmless, or when they're a mutation, potentially pathogenic. Okay, so an example of a missense mutation, and this is a very common one in the FKRP gene, which causes LGMD2I, and this is a founder mutation, and most patients in the US carry at least one copy of this mutation. Okay, so at base pair 826, a C changes to an A, which changes the amino acid at position 276 from leucine to isoleucine, L to I. Okay, now we can also sort of add or take away bases in DNA. So we can have an insertion. We just, stick, in this case, 3 more DNA bases. Now, you'll notice that because we do 3, and there's 3 DNA bases in a codon, it messes up a couple of words, but the rest of the sentence is still comprehensible. So, again, depending on, putting a couple amino acids that aren't really supposed to be there, that, may cause a problem, but the protein may still be partially functional depending on the exact situation. Okay, so for an example, okay, so here we, this means between base pairs 3505 and 3506, we insert three more bases, CGC, and that, adds between amino acids 1169 and 1170 an extra R. So it's, it's got one more amino acid stuck in there that isn't really supposed to be there. Okay, now what happens if we add not three, but just one DNA base? You'd think, well, okay, you're only adding one. That can't be as bad as adding three. No, it's much worse than adding three. Okay, why? Because every 3 letters is assumed to be a new word. If you add another C, then the reading, what's called the reading frame, the grouping of the 3 DNA bases into a codon is all wrong, and it's a mess. And if you make a protein at all, it'll be a mess. Okay, so so here's an example as it would be in a genetic report. Okay, so at after base 3505, you stick an extra C in, which means, okay, it does a couple of things. It changes that amino acid usually because it's a different codon. And then what typically happens downstream is that you typically will run into a stop codon, a new stop codon, because you're grouping things in the wrong way before very long. So remember, there's 64 possible codons. 3 of them are stops. So on average, if you just sort of choose random codons, you'll run into a stop after about 20. And that's kind of what here on the 6th codon downstream in the wrong reading frame, you run into a stop. So usually a frameshift is almost always pathogenic. Okay, you can also, well, a special type of an insertion is just called a duplication, which means that you just, repeat, a few letters of DNA, and so, but essentially that's just a special case of an insertion, so I won't spend a lot of time on that. Here we've added 3, so it doesn't mess up the reading frame. And a real example here, this is called a duplication, but it's really it's really functionally the same as an insertion. That just means that, the base right before the A that got stuck in, so base 1392 was probably an A, and then you stuck another A in, which again, causes a frame shift, and eventually you run into a stop codon. Okay, you can add in extra DNA bases. You can also take them out. Okay, so here would be an example in our sentence. If you just leave out the word rat, it doesn't really mean the cat saw the butt, not the bat. Okay, that doesn't make a lot of sense. So that's, depending on what DNA, what amino acid in a protein you leave out and what the structure of the protein is, sometimes that can, be, more or less functional, partially functional, or completely non-functional. One well-known example is the most common mutation in cystic fibrosis is an in-frame deletion. So essentially the mutated CFTR protein is missing one amino acid. Okay, and so this is an example from the calpain-3 gene that is a larger deletion. And this is actually one, you may remember in the talks yesterday, you heard that calpain-3 mutations can have either recessive or dominant inheritance depending on the exact mutation. This is one that's actually dominant. So this, you leave out 15 amino acids starting from, DNA bases, starting at 598 through 612. Okay, so 15 is a multiple of 3. That means you leave out 5 amino acids. And it's thought that those 5 amino acids are important to know to tell the calpain-3 protein, when to be active and when to not be active. And basically the calpain-3 protein's job is to chop up other proteins when needed. And if it doesn't have these amino acids, it just goes chopping up things at random times, and that's not good. Okay, the last type of mutation I'm going to go over has to do with splicing. Okay, so what is splicing? So the way that a gene is actually structured, so the coding regions are referred to as exons, which is why you have the exome is all of the exons, all of the coding regions in your genome. Okay, so but the exons in most genes are very widely spaced, and most of the length of the gene is not exons. Usually it's only 1% or 2. And splicing basically cuts out all of the parts of the DNA to concatenate all of the exons together, and then that gets translated into the amino acid sequence. So you can think of it as kind of like making a movie. You shoot each scene, would be a different exon, and then later back at the studio, they splice them all together and make a movie. Now, just as with a movie, some genes can actually be spliced different ways and make different versions of the proteins that include or don't include different regions of the gene. Okay, so we'll illustrate in our sentence example. Okay, so say that the cat is exon 1, and then and that's exon 2. Okay, so we've got a non-coding region which is called an intron, and then it has some DNA letters at the very beginning and end of the intron, which mark the place where it's supposed to be spliced. Then, okay, so what happens if, okay, so after splicing, you get rid of the intron, and then we get back to our sentence, the cat saw the, etc. Okay, what happens if we make a change in one of these sort of markers, in the splice begin or splice end sequence? Then the cell doesn't realize that's where it's supposed to splice. It just, keeps going and thinks that all the, DNA letters in the intron are part of a codon. Okay, and you get a mess. Okay, so the numbering that is typically used in genetic reports is the coding of just the DNA bases after the splicing. So the way that you indicate a DNA change in an intron is if it's near the beginning of an intron, you take the nearest end of the exon and put a +1. So this would be you delete a DNA-based gene that's the first base of the intron. So it's the base right after number 855. So 855 would be the end of an exon. And this would be near the end of an intron, just before the beginning of an exon. So here the beginning of the exon would be 3349, and then you count backwards 2. So that would be 2 bases back in the intron, and that would change from an A to a G, and that would also mess up splicing. So if you're reading a genetic report, the easy way is if the numbering contains a plus sign or a minus sign, that means that the DNA change is actually part of an intron, which usually means that it affects splicing. So that would be a splice. So if you don't follow all of that, just look for a plus or minus in the numbering, and that means you've probably got a splice mutation. Okay, I explained a little bit of this before. So in most genes, and actually in all of the limb-girdle muscular dystrophies, they're on genes where we have 2 copies of the chromosome, which means we have 2 copies of the gene. If it has dominant inheritance, then just having 1 copy will cause a problem. If you have recessive inheritance, then having 1 copy okay, and the other copy with a mutation, that means you're a carrier, but you don't have the disease. Okay, so for the other term you'll see in genetic reports is a homozygous mutation. What does that mean? That means that you have the same spelling mistake in both copies of your gene. A heterozygous mutation means you have a spelling mistake on 1 copy of your gene, but not the other. Now, what if you have 2 different, 1 spelling mistake on 1 copy and a different spelling mistake on the other copy? That's called compound heterozygous. Okay, so to just review, some of the words. Okay, so a variant just means any change in DNA from the standard sequence. None of us are the standard human being. We've all got variants. Most of the time, they're polymorphisms. Those are what make us all different from each other and unique, harmless. A mutation potentially causes disease. And vary of uncertain significance means we're not quite sure if it's a mutation or a polymorphism. So then some of the vocabulary. Okay, so a substitution in DNA just switches 1 DNA base for another. You can delete bases, add in extra bases, insertion, or as a special case of insertion, just duplicate the previous base or series of bases again. And at the protein level, a splicing mutation means that the exons aren't able to be spliced together correctly to make the final protein. A missense mutation means that you have a change in 1 amino acid for another in a way that causes a problem. A nonsense mutation means that you have you change a codon for an amino acid by a stop codon, which means you stop making the protein sequence before you only make part of the protein. And a frameshift means you aren't grouping the 3 DNA bases into the codon properly, and usually you get random nonsense. Well, I was going to say random nonsense, but why should I not say nonsense? Because in genetics, nonsense has a very specific meaning, so you need to be careful. That's the end of that, and don't know how we're doing on
we have 20 minutes left.
Okay.
It's going to wrap up at 11, so I guess, yeah.
Yeah. So,
well, first of all, thank you very much, Brad, for me talk. So I guess the great thing is now we have plenty of time for questions. So I think there's going to be a mic runner, or otherwise, if you can just project your voice.
I will say that I actually posted a version of this as a series of 3 videos on YouTube. And so, if you can actually review this on the web if you would like.
Cool. Thanks, Brad. So, yeah, don't be shy of the questions, because a lot of the questions the audience may have, a lot of other people may have the same type of question too. So, fire away.
Hi, I have a question over here. On one of your slides, you talked about inheriting dominant or recessive, and then what the effect might be. I'm wondering how it works that 4 siblings from the same family, only 2 of them are affected by a mutation. So it's a recessive. It's a recessive mutation. Why are only 2 of the offspring affected by it?
Okay, so if for a recessive mutation, you only have symptoms if you have mutations in both copies of your gene. So that would normally mean that both parents are carriers. So each parent has 1 good copy and 1 bad copy. So, from each parent, each child can inherit the good copy or the bad copy. So statistically, there's a 1 in 4 chance that you, flip tails twice and inherit the bad copy for both, from both parents. Now, that's the probability, but what happens in an actual family, you can flip tails several times in a row. So statistically, the family got a little bit unlucky. But I'm an only child, and I had a 1 in 4 chance. But I got LGMD 2B.
Hi, thank you for your great explanations. On a couple of your slides, you mentioned deletions and duplications, but they were fairly small. I'm wondering, in limb-girdle, are there occurrences of large duplications and deletions, the sizes that we see in Duchenne or Becker?
Yes. In fact, I have one. What is a large duplication or deletion? Usually that means that you're missing an entire exon. And yeah, okay. So you see like this one of these like red portions in the genomic DNA part in the upper right-hand corner. Say if you just like chopped out, in the middle of two white, portions, around an exon, and then just stuck the ends together, you're missing an entire exon. For various reasons, that tends to happen a lot in Duchenne muscular dystrophy, in the dystrophin gene. It's less common with in most types of limb-girdle muscular dystrophy, but I'm actually missing exon 52 of the dysferlin gene, and it turns out that there are several other people that are also missing exon 52, but we don't all seem to have the same boundaries of the deletion, which means that we're probably not all related to each other, so it's just a DNA mistake that's prone to happening every so often. You can also have a large insertion. Usually means that you're basically sticking in an entire exon twice. Is everyone sort of clear on that?
I'm just going to add to that point of Brad explaining it, but talking about the technical limitations. So deletions and duplications using, so the workhorse that a lot of clinical genetics labs and also research labs use is called next generation sequencing. I won't go into the technical details of it, but things that occur, what we call heterozygous, as Brad had explained, is very difficult to pick up in next generation sequencing, mainly because the breakpoint, so where it actually happens, the exact place it happens, is typically not in the exons itself, it's actually in the introns, so in the flanking regions, which with next generation sequencing, whether it's exome sequencing or panel sequencing, we don't capture that region. So we're left looking at coverage, and the problem with coverage, seeing how many sequencing reads we have in that area is it can be quite variable. So sometimes when we get half the coverage than we expect, which would sort of like correlate to a deletion, or if we see sort of like 50% more coverage, which would correlate to a duplication, sometimes we don't know if that's just variation rather than something that we're actually seeing in the patient's DNA. So some of those things are very hard to pick up with next generation sequencing. Other technologies are actually better to pick that up. And so sometimes patients go undiagnosed, they find one pathogenic hit and that's it. It's because that extra thing was harder to detect. That's an example of something that's harder to detect. So if you remember from my answers yesterday, sometimes people are undiagnosed because there's a challenge in detection, sometimes there's a challenge in interpretation. So these smaller duplications and deletions of one exon or a few exons that we typically see in limb-girdle, they're a lot harder to detect.
Yeah, that actually happened to me. I have one duplication of a few base pairs which showed up on my initial DNA test, but I only had one mutation, but I clearly had LGMD, and then only when they went back, and developed a test to look for large duplications or deletions did they find my second mutation. Now, for DMD, it's actually much easier to detect deletions because males only have one copy of the gene. So if you have a duplication, then you're getting no signal at all from that, from that exon. So you can, okay, where'd it go? And oh, it must be a deletion.
Yes, I just need some clarification. In autosomal recessive, you have two bad alleles, or a gene that's producing a protein. In autosomal dominant, you only have one bad gene. But the thing is that the DNA enzyme is coming down your gene and transcribing both genes in autosomal recessive and autosomal dominant. So one can only assume in autosomal recessive you're producing, two bad proteins that are non-functional or accelerating cell death. In the autosomal dominant, again, the DNA enzyme is coming down and transcribing one bad, but the other one is good. And I assume the bad protein is still eclipsing the function of the good protein that you have left, which is either gumming up the works, causing cell dysfunction or early cell death. Is that true? I mean, these proteins can be stopped early, and of course you have a small protein, but it really, autosomal recessive dominant are almost, just, I guess, archaic terms. It's really the protein that's bothering all of us today.
Yeah, so generally, okay, in genetics, there are always exceptions, is one important thing to be aware of. But most of the time in autosomal recessive diseases, they result from making a protein which either has a partial loss of function or a complete loss of function. Meaning that if you still have one copy, the protein made by one allele still functions, okay, you're a carrier, but you don't have the disease. Most is a dangerous word, but anyway, a lot of dominant diseases are associated with a gain of function. Means that the protein made by the mutant allele does something that it's not supposed to be doing, which causes a problem. So even though you've got another, the protein made by the other allele is doing exactly, if one is going crazy and causing problems in the cell, you still have a disease, usually.
Cool. Just before the next question, I'm sorry if you already touched upon this, Brad. There's another concept, sorry if you already touched upon it, called haploinsufficiency. So with the dominant cases, you're not producing enough of the protein because one copy just doesn't produce any protein, and you only have one then good copy of gene producing protein. Yeah. And so for some diseases, you need enough. Otherwise, you get a disease. So it's another mechanism in dominant cases. Yeah,
and one example then, this is actually due to having too much Down syndrome is caused by having 3 copies of chromosome 21, and there's at least some genes that if you have 50% more of them than you're supposed to, even though the genes themselves are fine, just having too much of the protein, causes all the symptoms associated with that.
Thanks so much, Brad. I have a question. So Serena, as you know, has 2B, and so that means her dad and I were recessive carriers of that genetic mutation. But what does that mean prior to us? Like, does that mean it was somewhere in our family history? Because neither of us knows what happened, about our ancestors. We know immediate family and a couple of generations up, but beyond that, we don't.
Well, okay, so the original spelling mistake had to, have started, somewhere, back in your family tree, but that's usually pretty hard to pin down. It may just be that, that the two of you were the first people, in your respective families that happened to have kids with another person who was a carrier. I mean, as long as, you can, have multiple generations of carriers, but until someone has kids with another carrier and, one of the kids happens to inherit both mutant copies, they're never going to know that they're carriers, right? So.
Cool. I'm just going to quickly add to that response the other extreme of that. So something that happened in the recent past in your family history. There's the total opposite end of that are what we call founder mutations. So Brad had a good example of one in FKRP. So there are particular mutations that are found commonly amongst some of the patients as either one of their mutations or both their mutations. And these are founder mutations because, one, they happen a long time in the past in human history, and then the ancestors of that person just happen to pass, was passed on more kids than expected, and just by random chance it increases because the people who are carriers are either unaffected or, in some really, really rare cases, for some diseases such as sickle cell, that there is an advantage to being a carrier. But for muscle diseases, as far as I'm aware, there is no advantage of being a carrier. So it's more by chance that because it happened a long time in the past, that the frequency of that is a lot higher for patients with that disease.
So this may be a question for after lunch, but do we know which type of mutation is the easiest to fix or is being fixed now? I know they're all difficult, but is that...
Okay, it's Munkel is definitely going to be the expert. So, but I'll say that it sort of depends on what the fix is. Now, if it's gene therapy, or gene replacement therapy by AAV for a recessive disease, it doesn't matter what your mutations actually were. Because you're not doing anything with the incorrect copy of the gene. You're just giving the person a new correct copy. So it doesn't matter what their spelling mistakes were. Now, for things like gene editing, then, that's Munkel's department.
Yeah.
Jump in.
I can jump into that. So I think as Brad said, the best, the easiest, there is no easy, but the one that most research effort has been invested into is scenarios where you don't produce the protein at all. So if you're introducing a working copy of that protein, a healthy copy of that protein, there's no worry about what effects it would have on the cell. But, immune response put aside, okay, with that, which maybe Dr. Mendel and others after lunch will talk about in that case. Then there's a scenario of what I think Jeff Chamberlain has explained of dominant diseases where that's a runaway train. If it does something that it shouldn't be doing. So that's much harder to stop. And one way we can do that in dominant conditions is have a therapy that specifically targets that mutation and makes less of it. So it will make it less severe, and there are ways we can do that. And then there's therapies that look at skipping over regions with a mutation, and that's one of the first therapies that Sarepta tried out. So if you remember from Brad's presentation where he talks about things that are out of frame mutations. So it's a way of skipping over exons, the red things in that picture there, but still have it in frame. So you lose bits, but the sentence still relatively makes sense after the bits you skip over. So you can skip over mutations. And the last thing, which is an exciting development in terms of CRISPR, is being able to change letters. So there's only a subset of letters we can change, but go and change a letter. If it's just a one-letter change, that's your mutation. So, but answering the other part of the question, are there things that are still difficult? There are still a lot of types of mutations that we can't address. So it produces proteins, so we worry if we introduce a new copy that it may have an effect on the existing protein that you're actually still producing. But as you said, it would make for a good lunchtime conversation.
Okay, so I think we have time for one more question.
We've talked about recessive and dominant genes, but what about a new mutation? So if you have a new mutation, are those typically dominant or are they recessive?
Could be either. I'm not. Yeah.
Yeah.
It all depends on the mutation and the gene. I would think that it's probably, if you just sort of randomly start making changes in DNA, it's probably a lot more likely to make something non-functional than have a gain of function. But there are dominantly inherited diseases that tend to have recurrent mutations. Munkel, you?
Yep, so I can jump in and try and answer that. So from human genetic studies, we know that new mutations, which we call de novo mutations, so mutations that parents don't carry and a child carries, so there's been a mutation in either the sperm or the egg, and that has actually caused that, either a subset or all eggs or sperms, and then that gets passed on to the other generation. That happens, I think, you get about 1 to 3 of these de novos per generation, or 0, so 0 to 3. And so just because there is a de novo mutation in a gene, it doesn't necessarily mean it's causing a disease, like with the variants of uncertain significance, rare variants we see in the population. But what we do see is they tend to be dominant rather than recessive, because for it to happen, to be a recessive, you'd have to have a new mutation in one of the parents and the other parent being a carrier. It's not to say it doesn't happen, but the dominant version of that actually happens a lot more than the recessive version, because one has to be a carrier. Because they happen in such low frequencies, to have both parents being de novo and then passing it on also with a 25% chance to affect a child, it's just a much lower probability event, just going through all the different scenarios.
Yeah, well, with again, as there's always exceptions in genetics. DMD is an exception that, if a woman, has a de novo mutation in one copy, then any sons who inherit that copy because they only have one X chromosome, have the disease. And just for reasons I won't go into, the DMD gene is very prone to de novo mutations. Cool. So, well, hopefully this has been helpful, and you can dream about rats and cats and bats. And so thank you all for coming, and both to the...
Thank you.